4 a.m. is the best time to plan out experiments. More specifically, 4 a.m. the day before your experiments. Even if you don't want to take that piece of advice, here's some more advice on planning out experiments and actually performing and the experiments and doing the analysis afterwards. So actually, I'm working from home this morning because I find it helpful to concentrate and really plan out these big experiments. So I have to do a big experiment tomorrow with lots and lots of replicates and lots of stuff, and I really don't want to mess it up. So I want to take a lot of time beforehand in order to concentrate and really lay everything out. And I'll show you how I do that with like spreadsheets and various things, so that during the experiments you can just focus on the experiments and not like, oh my god, did I calculate that wrong, or oh, I needed to add this control. So you want to really think about these things ahead of time. And so let's take a look. So there are things that you should do before the experiment, things that you can do during the experiment, and things that you can do after the experiment to make your experimenting experience more enjoyable, as well as your future you will thank you when you look back at your notes and see that you actually took good notes. So it's important to really plan out your experiments. And so when you have big experiments, like tomorrow, I need to do these really important experiments where I've been waiting for this protein to express, and I finally have this protein, and I'm going to do these kinase assays. And you don't need, I have a post on this if you want to know more, but the basic idea is I hope you can see from this that there's a lot of samples and a lot of work and a lot of places where things can go wrong. And this is just like the final part of the experiment was you have to prepare all those samples in the first place. And so there's a lot of things and it's really stressing. It's really stressful. And so I like to plan things out really well ahead. And I like to plan things out the day before. And then the day of, I can kind of review them and have with a fresh set of eyes, and hopefully everything still looks good. So when I'm talking about like planning things out, I'll show you in a minute, but I seriously plan things out because my brain, when I'm experimenting, I just want to be able to focus on following the instructions that I've already written down for myself. So I literally write out like how much I need to pipette where and when, how I'm going to order my samples, everything. And so let me just show you an example of what I was doing this morning, but I've taken out the information about my actual experiment, but just the basic overview. So when I do experiments where I need to do calculations, where I have a bunch of samples, all of these various things, I keep a spreadsheet, and I have like a directory where I have a bunch of folders that all have the experiments, and I label the file by the date name and then like the experiment type. And then all those experiments, I have like a folder for like the setup, this folder for the calculations afterwards, everything very organized. And one just word of caution is to make sure that when you're copying and pasting between different, like if you're doing a similar experiment on a different date, make sure that you change any information that you need to change. And because this can cause problems if you have old information on there from a previous experiment that you just copied and pasted and isn't still really the case. And also that's another reason why we'll get into that you should really be taking good notes, so that if something was wrong with one of your calculations, you then don't care. I like to, if something was wrong, then I make it red. I color it red after if I realize something was wrong. And that way I don't carry that mistake on into future editions. But so I have the date, I have the experiment type or overview. So basically this would be like kinase experiment, blah. And then the goals, why am I doing this experiment? What if sometimes I put like, this is a redo of an experiment I did this day, but there was a problem that day, or that sort of thing. Or this is an extension of this experiment I did this day, but I wanted to test this in addition, or that sort of thing. Then the samples I'm testing and the concentrations and reaction conditions. And then I do the protein preps used. So when I do a protein purification, then I put on the tube and stuff what the date was, and then I will be able to look back and see, if I'm having a problem with an experiment and I'm using the same protein prep each time, to have this problem with a different prep and that sort of thing. So I have a whole database of these preps, and that I keep updated when I have new preps, and also like cell pellets to purify. But basically I have a lot of databases and a lot of notes, keeping track of things. Then I'll have my various samples that I need, the things I need to prepare. And so I have this is basically my summary tab. I have other tabs where I'm actually doing the calculations. And so I have tab set up with like the starting concentrations, the desired concentrations. I can change things and then it'll do the calculation so I can figure out how much of these various things I need to add. And then I can copy and paste it into my summary tab. And remember that with copying and pasting with Excel, you need to make sure you're just pasting like the data values. And so, or else it's gonna, if you're pointing to a row, it's gonna get all confused and give you one of those reference errors. But so basically I do this for each of the different things I need to make. I actually have a lot more to make, but I took them out. And then I have basically all my sample tubes where I need to, where everything was going to have in here, what it needs to be in here. And then this is for this insulation counter, where what the different samples are. And so I can cross-reference later. So again, there would be I have different tabs where I was actually doing all these calculations, but then I want to have just like an overview, because a lot of times what I do in my notes is I actually just like think things through. So I'll write out like my thought process so that later I can look back and be like, oh, that's why I did that, instead of like, why the heck did I do that? So I write out my thought process, but then the problem is I can have a bunch of numbers all over the place and it can be too extensive. And when I'm doing the actual experiment, I want to have something really simple so I don't have to think. I can just pipette here, pipette this here, pipette this here, pipette this here, pipette this here. If I'm doing something with time points, that sort of thing, I'll have a schedule. So I'll say, okay, at this time I need to have done this, at this time I need to have done this, at this time I need to have done this. Especially helpful if you have like staggered time courses and say, okay, at this time I was do this for this sample and this for this sample. So yeah, so you kind of have to find what works for you, but you want to have something really just like balanced. So I'll print out these experiments. So I make these ahead of time and then I print them out. And then, so when I'm in the lab, I have access to them to look at. I don't like to do things digitally when I'm doing these types of experiments, especially when I'm doing radioactive experiments. I'm not taking my laptop in there with me. I keep a digital notebook and a physical notebook, and then I take pictures of my notes every day. And then I put them into my electronic notes as well. And then I copy and paste from my Excel file into my electronic notes. So everything's all connected, but I have everything in a centralized location so I can find it. We have everything set up so you don't have to think during the experiment. You can just focus on the experiment and not get like panic that, oh my God, did I forget something? Speaking of forgetting things, you need to make sure that when you're setting up your experiment, you have everything that you need. You've designed your experiment so that you have all of your necessary controls. So this can be a big problem if you do this experiment, but you don't have a control. And so you don't have, you need some specific control. Like that's why I have so many samples. I only want to test like a couple things, but I need all these controls in order to show that the thing I'm testing is really the thing I want to test is really the thing I am testing. And it's not like an effect of something else. So it's not a different variable that I hadn't controlled for. And so I have a lot more on controls in a different post, but you want to make sure that you really think through what controls you're going to need, because sometimes you do this like huge experiment, you do all of these various things, but if you forgot your control, well, now you have to redo everything with that control. And so it can be a lot of work. And so a lot of times there are multiple controls. And if you think you've controlled for everything and then you realize you haven't. So you don't want this to happen to you. So make sure to include those controls when you're doing your calculations. So now that you've done everything, you need to make sure that when you're actually getting ready to start the experiment, you have everything that you need nearby. This is especially important when you're working with messy things or things like in a hood or in a hot room. So with radioactive stuff, with things in a room that you're not used to, maybe you're working in someone else's lab because they have a piece of equipment you need or that sort of thing. Make sure that you have everything that you need and you don't have to like panic. Oh my God, where is this? Where is this? Also identify potential wait steps where you can like do things like go to the bathroom or get coffee. Sometimes when you have experiments and it seems like you can't even get any break, if you have like, like, okay, there's going to be a wait, there's going to be 5 more minutes, I can make it to 5 more minutes and then I can get this little break, and walk it through in your head. So I like to kind of walk through my experiments in my head when I'm planning them out, and then like before I actually start to do them. So now you're ready to do the experiment. I have this problem where when I'm focusing, I hold my breath. And so I have to remind myself to breathe. Sometimes I'll be doing these experiments and like all the wells are starting to like merge together. And I'm like, oh my God, I can't think. And I have this headache. And I'm like, oh, I'm not breathing. So breathe. So periodically check to see if you're holding your breath. And if you are, stop it. And use your planned out notes. Unless you run into difficulties, follow your plan. Remember, your brain was working better when you were planning, and now your brain is stressed and exhausted. And so you want to make it even easier for your stressed, exhausted brain. I like to color code and number code, so everything is easy to find what I need, where I need to put things. When you have a plate like this, it can be really confusing, like all of these wells look identical. But if I'm able to color code them, then I am able to know, okay, this is a different well than this is. And then like ABC, you make sure you keep track of things, so that you don't do all this work and then not know what it corresponded to. Also, okay, so when you're doing your experiments, you want to make note of anything that was strange or unexpected or any deviation from the plan. So this could be something like it looked kind of cloudy, or I incubated it for 35 minutes instead of 30 because I had to wait in line for the centrifuge, which also is a benefit of getting up early, is you don't have to fight for the equipment as much. But yeah, so take good notes when you're doing your experiment so that you can look back and know if anything was different. And then when you're doing an experiment, like I like to imagine what the molecules are doing. It helps you remember the purpose of the various steps, and it's fun. So sometimes these experiments can get really, really boring when you're doing the same thing like over and over. So in addition to like podcasts and stuff, imagining what the molecules are doing can be really good. Yeah, some experiments you can podcast doing, some experiments you can't. Some experiments, like parts of it, I really need to focus. So I have like noise canceling headphones so that I can really, really focus. But then for other parts where it's just like manual work, then I can like put on a podcast and kind of like half focus to it, but like keep on to my experiment so I don't get too distracted. Okay, so now the experiment's over. Now you can get coffee and water and go to the bathroom. Like, I know I keep saying that, but it's like a serious thing. One of those too busy to pee experiments. And so then you want to write down your thoughts while everything is still fresh. So your brain is going to be super duper exhausted sometimes if it's one of these long experiments. But the future you will thank you, present you, if you write down things while it's fresh in your head. And so sometimes you'll see in my notes, I just have these like random like brain thoughts, like blah, blah, blah. I mean, like my notes aren't like cohesive at that point. But I have my thoughts on paper before I call it quits for the day. And so that way, if there was sometimes if like there was a weird sample or something, you want to write that down before you forget. All of that stuff. Sometimes I have it written down in my physical notes, but I haven't typed it up yet. But as long as you get it down, I take a picture of my notes. I can then type them up, even the next day if I'm too tired then. And so, of course, you need to clean up your experiment. Part of the reason why radioactive experiments take so long is because you have to really, really clean up, make sure you check everything for radioactivity, all of this stuff. But yeah, so the future you and your colleagues will thank present you if you clean up. So sometimes it's like, oh, I'll just clean up tomorrow. But then tomorrow you're like, oh, why didn't I just clean up yesterday? And so I'm not talking about radioactive stuff here, just like recharging your column or that sort of thing. And so, yeah, so do it now, don't procrastinate. And then format your results for the clearest feature interpretation. So when I am doing experiments, I like to make a little like summary write-up type of thing, like in a figure manner. That I do this in Adobe Illustrator. A free alternative is Inkscape.
but it's a vector graphics software so that basically you can save things as PDFs, which is good because then you can blow things up and they won't get all pixely. But I like to save it in a way that I can crop it in different positions and then have different information. So often I'll have figures like this that I can then use if I'm doing a lab meeting or if I'm meeting with my PI, so my boss or that sort of thing. So basically it has all the information that you would see in like a paper figure, but also some more information like more experimental details, as well as the date. And this way you have this data to go to nicely formatted with all the information that you need without having to dig it up. And then I actually have in my electronic notebook, I have a results tab. And then in my results tab, I have broken up by the type of experiment. I then have PDFs of my experimental results for different days. And I title the file when I save it, I put like the date and then like the experiment type so that I can search it. The date is like super duper important, especially if you have good lab notes, then you can cross-reference back to the date. But this way you can then like go to find the experiments and present them however you need to, and you can crop them out in different ways so that they'll do things. So this was a major benefit of learning Adobe Illustrator. And so speaking of this stuff, try to get at least one presentable figure from an experiment before you move on from a dead end. Because like, as you found when you were doing, when I was writing my thesis, is that sometimes like there was a project that you ended up abandoning, but you didn't have a good figure to show like, this is why I abandoned it because it wasn't working, or it show this one thing, but then you don't have like that one figure that actually has everything in one place. You just have a bunch of different figures, and then you might have to redo an experiment that was kind of pointless just to show that it was kind of pointless. So it is not, don't worry about having things that you need to like directions that you abandon, but make sure that you have something, this like one figure that just like shows why and how. That was something I learned when thesis writing.
So you can get into some problems if you basically just focus on just doing experiments all the time. And your brain starts to feel like your lab bench looks and you get really burned out. I had this problem early in my PhD where I just felt like I needed to be constantly experimenting. And I just went from one experiment to the next. I didn't really have time to really think about the experiments more and plan them out better ahead of time, write more about them afterwards. And I still have this problem where I feel like this kind of like guilt whenever I'm not actually doing experiments. So last year I had this post, I'm like, is it okay to just like take a day to read papers? And I did, and it was awesome. And I have tips on how to read papers and that sort of thing. But the point is that you don't need to always be experimenting. And when you are experimenting, you want to make sure that you're maximizing the potential that your experiment is actually going to give you the results that it's going to give you answers. So those might not be the results you were expecting, but even like negative results are still results. But you want to have results that you can trust, and that experiments that you won't have to do more times than you need to. And so earlier on in my PhD, that was a really big thing for me, was realizing that instead of rushing from experiment to experiment to experiment, take some time to really plan things out. It's hard when you have like an idea and you're like, oh, I just want to try this. But if you don't try it the right way, then it's not going to be helpful at all. And you're just going to have to redo things. So try to curb your enthusiasm and really plan things out before you dive in.
So I know that the whole morning thing isn't for everybody, but I really am a morning person. And during the pandemic and lockdown and everything, I found that I could focus a lot better if I get up early and work from home for a bit. So I got permission to work from home today so that I could focus on planning the experiment and doing some writing for my paper. And so I was able to plan out my experiment. I still need to check it again and then again. but I was able to plan it out and get some writing done. And now I'm taking a break before I have a meeting in a little bit. But super productive morning. And so, yeah, morning. My brain works best then, but I crash at night. Anyway, so, and I have a page on my blog with a bunch of posts on various types of experiments, but this was just like a post on how you prepare for these experiments, whatever the experiment type. And yeah, so happy experimenting.